but it's evolved a particular unique function. And the function that this has, its biological function, in this case to break down this nasty toxin, is directly related to the specific structure, this fold that the chain has taken up. And there are lots of these stories throughout biology where we can find interesting biological functions and interesting adaptations that organisms have made. And if you look on the molecular level, there are proteins, and those proteins have unique three-dimensional structures to them that are responsible for conveying those biological functions. So there's this wonderful correlation between biology, sort of something, a particular function that something has, and molecular structure on the other side, these beautiful three-dimensional shapes that proteins adapt. And so I'm a chemist. I'm interested in the structures of molecules. And so for me, these are wonderful problems to study, because if we can learn more about these beautiful three-dimensional structures, we can begin to come up with descriptions in more detail of some of the biological functions. So a big question is, so if you have a protein, and it comes out like this wet noodle, this wet piece of spaghetti, okay, and that's what we call sort of an unfolded protein. It somehow has to fold into one of these really gorgeous three-dimensional shapes on this side. And in a sense, this is just a chemical reaction, right? We have this floppy protein here, and it has to kind of adopt and fold into a unique three-dimensional shape. And it does this automatically. It doesn't need assistance from any type of machine, though there are machines available inside the cell to sort of speed it up. But they aren't really fundamentally, they aren't intrinsically needed for this reaction to occur. It happens spontaneously. If you take an unfolded protein and you sort of let it sit on the lab bench, it will fold up into the right three-dimensional shape. And you are actually folding proteins inside your body literally thousands of times, every few seconds, okay? Because there are cells inside your body that are producing proteins, and they are folding. And you aren't thinking about it, it just happens. Okay? What's remarkable is that from the perspective of a chemist, this is a very difficult reaction to describe, and we don't really understand, from a chemical perspective, how this happens. How do you get from here to there? And that's called the protein folding problem. Okay? It's sort of a misnomer. As I said, you fold proteins all the time without thinking about it. It's not really a problem for your cells in terms of how the proteins fold. It's a problem for chemists in understanding, what's the fundamental chemistry that dictates that this wet noodle forms this specific three-dimensional shape. Now, I got attracted to this in part because this is a really fascinating physical chemistry problem. And my interest in chemistry just led me to really wonder a lot about this, and it's led many, many others out there to wonder how this happens as well. But there's another reason this is interesting, and it has to do with this down here. And I guess I'm wondering, how many folks have made Jell-O? Has anyone made Jell-O before? Okay. So there's a kind of two-step procedure usually to making Jell-O. You take the stuff out of the package, you put it in boiling water, you dissolve it up, and then you stick it in the refrigerator, and it kind of undergoes this transition in the way it looks, and it makes the sort of solid Jell-O-like shape. So when you're making Jell-O, you're actually doing a protein unfolding reaction, because Jell-O is made up of largely a protein called collagen. It's a little bit gross where they get the collagen from in the Jell-O factory, but if you're curious afterwards, we can talk about it. So it's made up mainly of this protein collagen. Collagen has protein chains that are wrapped tightly in a sort of sharp helical shape around each other. And when you heat it up, what you do is you separate out that helical shape. So the chains begin to fall apart and separate. Now, when you heat it up, you boil it, you break apart the protein, you unfold the protein. You take it from its stable shape to this shape. Now, what happens is, when you cool it back down quickly, instead of going back to the correct shape, it goes down this alternative path, and the chains get all tangled together, and they make something that's Jell-O-like. Okay? So you unfold the protein, they come back together, and they associate into the wrong shape, or they misfold, and that misfolding leads to what we call an aggregate. All of the chains tangled up together, and that aggregate is what gives the Jell-O its consistency. Now, this is fascinating, so that, in your body, you are folding thousands of proteins, every few seconds. Almost all of them fold correctly. once in a while a protein will fail to form the correct shape. Okay? And that unfolded protein, if it gets together with other unfolded proteins, can begin to form protein aggregates. And these protein aggregates, when they accumulate inside your tissue, it's like making jello inside your tissue. And that's not a good thing. And there are many, many diseases which are associated with the misfolding of proteins and the accumulation of aggregates. So the protein folding problem is interesting, partially because it's this intrinsically cool chemistry problem to study, but partially because when it fails to happen, and it's not very often, your body's very good at folding proteins, but once in a while, when it fails to happen, the consequences of that can be very, very serious. And so if we can understand more about how proteins fold and why sometimes they misfold, we can begin to understand some of these misfolding diseases. So misfolded protein, a whole class of misfolded protein fall into the category of what's sometimes called amyloid. Amyloid is a term that sort of dates back from the 19th century of these kind of deposits that could be stained if you took certain tissues, if you took tissues from a patient who had died from any number of diseases, and you sectioned the tissues and looked at them under a light microscope, pieces of it would stain into these dark patches that were identified as amyloid. They were sort of these protein-rich deposits that you could find. And so this is, for example, an image of brain tissue from a patient with Alzheimer's, and you see these very darkly stained patches here, and that's accumulated protein, okay, that's existing outside the neurons in that tissue. Now, if you were to zoom in and get a very high resolution picture, so right now this just looks like a glob of protein that's there. But if you zoom in more closely, you find that that protein is actually, that glob of protein is made up of all of these fibers. Okay? And these fibers are about, say, a few nanometers wide. And so a nanometer is a thousandth of a millimeter of a human hair. So, a couple of nanometers wide. And they can be many microns long. And micron is about the width of a human hair. And so these kind of tangled up fibers make up these blobs, these jello-type blobs that you find in tissue. And they can be found, there are a lot of diseases which are associated with the accumulation of amyloid. Alzheimer's disease is one of the more famous, and it's the proteins called A-beta and tau, which are found in these tangles in the brain tissue of those patients, but also other neurodegenerative diseases, such as Parkinson's disease and Huntington's disease, and Creutzfeldt-Jakob disease. It's a spongiform encephalopathy. It's the human version of what's commonly known as mad cow disease, okay, which is made up. It's a disease where you get accumulation of these protein tangles in brain tissue. But interestingly, there are other diseases as well that are amyloid diseases. Type 2 diabetes. Turns out that if you look at tissue from the pancreas of a patient with type 2 diabetes, you see protein tangles which look very similar to the protein tangles that you see from the brain of a patient with Alzheimer's disease. Okay? Different proteins involve different tissues, but the formation of amyloid is something that these diseases have in common. So there are a lot of diseases which are related to forming this amyloid. And again, this amyloid is a product of sort of a different path that a protein can take while folding. Okay, so instead of going to a folded state, it actually ends up in one of these tangled states or aggregated states. Now, we can draw a cartoon model here, where if you have a protein that somehow is in what we call an amyloidogenic intermediate, so it's kind of caught into some shape that's not its final stable state. These amyloidogenic intermediates can begin to stick together, they begin to form fibers, and these fibers eventually lead to deposits. Now, as a physical chemist, I'm interested in a few things. One is understanding things in terms of their molecular structure. So can we draw pictures of molecules with atoms and bonds and detailed structure instead of just cartoons in these cases? And the other is really trying to understand details of the energies and physical interactions involved. So can we explain what are the forces that are responsible for each one of these steps once we understand the detailed molecules? And that's really the goal of the work that we do in my research group, is can we take this picture that we have these chains that get tangled up and make jello, and can we begin to think of those as detailed molecules and understand the chemistry of that in with a more precise picture.